Hello, everybody. I'm Azari Guerrero. I go to Telluride High School, and this summer I interned at the Burton Lab, which is part of the Immunology and Microbiology Department at Scripps Research in San Diego, California. Okay. So a little intro before I dive in. The Burton Lab specializes in antibody discovery and characterization against viruses and infectious diseases. For those of you who don't know, antibodies are proteins produced by our immune system to recognize and fight off any harmful foreign substances. So I worked with Dr. Nathan Butler, who specializes in malaria research, and Dr. Benjamin Amos, who specializes in HIV research. I also worked with Shiloh, as you know, and her mentors, Sean Callahan and Nathan Liendo. So the main thing we did was antibody production, which essentially consisted of designing DNA that would produce a specific antibody, then DNA replication, amplification. Then we did what's called cell transfection, which essentially means infecting cells with the DNA that we designed, which would then produce our antibody of interest. After they were produced, we isolated them and purified them, then analyzed their behavior and potential using techniques such as gel electrophoresis, ELISA, luminescence plate reading, confocal microscopy, and sequencing. So pictured here is me and Shiloh's antibody solutions, and then next to that is the confocal microscopy machine. So I worked with antibodies specific to Plasmodium falciparum, which is the parasite that causes malaria. It did take me about a week to figure out how to pronounce that, by the way. I did get made fun of for that as well. So down here is the parasite itself, which I was able to look at through confocal microscopy. And then we also performed a challenge on mice where we infected them with malaria and then analyzed how each of our antibodies interacted with the parasite in vivo, which is pictured at the top there. The end goal is to find an antibody that permanently inhibits the parasite. Doing so would save the lives of millions of people worldwide, especially children under the age of 5 in Africa, who are the most vulnerable to the disease. So the thing that surprised me the most from my internship was how casual it was. I went in pretty nervous and intimidated, but I very quickly found that everyone there was fun and welcoming. Shiloh and I's mentors even invited us to hang out outside of the lab, which was very surprising to me. So they took us out to eat a bunch. They tried to teach us how to surf. And then after all that, they took us around San Diego to get to know the area. I think this casualty made the whole experience a lot less intimidating and more comfortable, which in turn allowed me to be very engaged and involved in my learning. So as you know, I was in San Diego with 4 other girls and our host mom, Jenny. We did a lot of stuff, as you can see. Yeah, we went to a different beach every weekend. My favorite were definitely Scripps Beach and Del Mar. We went to a few farmers markets around the area, and then my favorite part was definitely going to the weekend concert in Los Angeles. Please don't ask how much money I spent there. My biggest piece of advice for future interns, or my biggest takeaway really, is that your mentors are not going to expect a high schooler to immediately understand all of these complex concepts. Your mentors are there to share their knowledge and love for science with you. You're not there to give them the solution to all of their research. With that, there's no dumb questions. As long as you're asking questions, I think you're doing everything right. Also, make connections and don't take yourself too seriously. Science is fun, so it's okay to have fun with it. And thank you to all these people.
Awesome.